Hello everybody, I'm Frederik de Rose. I will be your host and moderator in this webinar. We have a very exciting agenda for you. So we have excellent speakers for today. The webinar will actually focus on hollow fiber assays, the challenges, and of course the specific focus in the use in antimicrobial drug development. So the webinar will be composed of several talks, and then we also have plenty of time to go to some questions. You might be able to use the chat to post your questions. In the interest of time, we will answer the questions, or try to answer the questions at the end of the session. So we'll first have the three speakers of today. Myself, I'm actually part of Connecting Pharma, which is part of the AMR Accelerator and Combine. So we are moderating this webinar, and I have the pleasure to have three excellent speakers. Professor Shampa from University of Liverpool. Then we have Professor Santiago Ramón-García from University of Zaragoza. Hope I pronounce it correctly. And then we have Professor Alasdair MacGowan from Bristol University. So welcome, the three of you. Very happy to have you on board for today. So what we'll do is we'll first start with the presentation of Shampa. And Shampa will be talking about why do we actually use the hollow fiber infection model in antimicrobial drug development, sort of as an introduction, and then we'll go into more technical details later on. So Shampa, a very warm welcome to you, and the floor is yours.
Thank you very much. Hopefully you can see my screen, the presentation on the screen. If not, please do give me a shout. So thank you for the introduction, and hopefully by the end of this you'll have some ideas about why we use hollow fiber infection model. So firstly, my disclosures, and just to say all the views presented are my own. So let's start from the beginning of, why do we use what do we need from our preclinical models in the first place? So one of the aims is we want to demonstrate some efficacy, and ideally we'll do this in animal models, in vitro models, and that gives us some idea that we're going to have something that works in the clinic. But something we have to consider with these models is how well does the outcome measure relate to clinical outcome. So, often in animal models, we'll have a sepsis model. We're looking at survival as an endpoint, and that's fine. It's telling us the drug's working, but it doesn't necessarily give us enough information to decide how we're going to use that drug, what dose we're going to do in the clinic. So the other thing that we really need to focus on is understanding a PK/PD target. So actually, we want to understand that pharmacokinetic, pharmacodynamic relationship. And from that, we want to derive an exposure target. Then we're saying if we achieve that exposure, we should see a positive clinical outcome from that. However, in order to do this, we need to understand this across a great number of strains. Quite often we're actually trying to develop something that could be broad spectrum. It has activity against a number of different families of gram-negative pathogens, for example, and we can't just do our preclinical data workup on, say, one E. coli strain or one Klebsiella strain. We need to understand a number of different strains from a number of different pathogen types and across a range of MICs. So it's a great deal of data we need to generate. And the other thing is we need to understand resistance. So it's not only sufficient for us to just look at bacterial killing or stasis. We need to understand, that our drug's going to work for a while and not just become resistant quite quickly. So, things we need to bear in mind also is the duration of the experiment. So traditionally, we've used for a long time animal models, and that they're still very important, but there are some limitations with animal models. Variability, just natural animal variability. Sorry, my builds aren't working here. Here we go. Is one of our challenges. So the data I'm showing on the panel in the right here is data for a compound that's been tested in both a thigh infection model and a lung infection model. And the driver here on the actual y-axis is free time above MIC. And this isn't a statement on this compound by any means, but just to give you an idea of the kind of variability that we can see generated from this animal data. So you can see on the left here, you know, we've got a number of different isolates from E. coli and Klebsiella, and actually we've concluded a free time above MIC can range from anywhere between, you know, 10% to 100% free time above. So you can get a variability not just down to the strains, but also possibly down to variability through your animals anyway. We've then got to consider things like infectivity. So what I mean by that is, you know, we're actually giving a strain of bacteria to an animal, but actually how that develops an infection and whether the bacteria grows is something we look at. Not all bacteria will expand in the animal model, so that can limit us. We're quite limited with duration with some of these models. You know, actually they're quite severe models. Quite often we're making them neutropenic in order to allow us to understand the PK/PD, and actually that has an impact on limiting the duration that we can run the experiments. Certainly we would lose the controls even if we could run them out with the actives for longer. So it doesn't really give you a good basis for understanding PK/PD. And that has an impact on us being able to understand resistance in these animal models. And the other thing is the ethics of it. So, you know, really, whilst animal models will continue to be important for some time, we need to be more considerate with how we use them. So there's that ethics and that 3 R's that we need to consider, you know, the replacement, the refinement, etc., when we're considering how to do this. So moving on now to the EMA guideline, and this is a guideline for use of PK/PD in the development of antimicrobials. And this has been around for quite some time now. I think it was last updated around 2017. And there's just a few things I'd like to point out from this that pertain to the in vitro models, where it's considered they can be used to describe PK/PD relationships. And it, you know, mentions here that you need to have representative organisms and a range of inocula. You can use these models to look at the effects of different types of PK profiles, which I'll talk about shortly, but also to study the relationships between rates of emergent resistance, drug exposure, and the duration of therapy. So what kind of models am I talking about? And although I've introduced this as hollow fiber, I actually mean dynamic in vitro models. And certainly from this slide, this is not exhaustive on the types of dynamic models that are available, but I'm only going to mention 2 that are relevant to this afternoon's set of presentations. And so the reason we call them dynamic is that they're allowing us to access activity following changing drug concentrations. So they're different from the static time kill model, for example, because here you can follow a full human drug concentration profile. So there's 2 models that I'd like to point out here, the one compartment model and the hollow fiber infection model. And really they both work on the same principles. So here you have a set of pumps and a set of different media. So you have drug-free media, a central compartment and a waste compartment. And actually the way the pumps and the drug media go in actually help you produce that humanized PK profile. In the one compartment model, the actual bacteria is grown in the central compartment, but in the hollow fiber infection model, an additional compartment is introduced with this hollow fiber cartridge. So in this hollow fiber cartridge, you have, sorry, this pointer's not working that well. So what you have a bundle of hollow fibers. So they're really like renal tubules that are used in dialysis. And so actually now the drug and the media are being pumped through the cartridge and through these tubules here. And what we're doing is we're growing bacteria on the outside of this. So this allows us to pump through a humanized profile, but also the way this is designed, it allows the exchange of drug in and out of those hollow fiber tubules, as well as the exchange of nutrient in and out as well. And so that media is continuously being replaced. So what are some of the reasons that we go and use these dynamic in vitro models? Well, there's one advantage in that it can remove some of that animal variability, but I don't want anyone to think that there's no variability associated with these models. That there certainly is from a number of different reasons. But you can increase the duration of the study. So quite a number of these models, you know, when we're looking at standard gram-negative or gram-positive bacteria, maybe we'll run the experiments out for 10 days or so. But in some in some areas you can run them out for a lot longer to really understand that both the effect but also emergence of resistance. You have much more flexibility around the inoculum size, so we can go in at a higher inoculum that helps us to understand resistance. And there's a bit less limitation on the types of isolates that can be explored. So more isolates that you put into these models are likely to expand. We have the advantage here of continuous endpoint measurement. So a lot of the times when we're doing our animal models, we're tending to look at bacterial reduction at a single time point at the end of the experiment. So for example, 24-hour models is what we typically use. So we're just looking at the 24-hour endpoint, although there are different designs as well, but they use more animals. So here we can look at the change in bacterial reduction over time. And with that, we can also measure PK. So you're getting your PK not only from within the experiment, but also continuous throughout the study. Remember, in animal models, that's quite often limited. Oftentimes, especially when using mice, you have to understand your PK in a satellite group of animals. So they're not the animals that you've seen the infection in. And sometimes that's done in infected animals, and sometimes the PK is done in healthy animals. So the biggest advantage here of the hollow fiber is that we get to mimic human PK. Now, it doesn't negate what we can do in animals, because certainly what we're trying to understand is a PK-PD relationship, and we can use our exposure measure and modeling of human kinetics to understand what kind of PK profile you need in humans. But still, there's advantages to being able to mimic the PK profile in an infection model to answer different questions. And I'll give some examples over this over the next few slides. Certainly doing an infusion in this model is a lot more practical than trying to do that in a mouse, although, you know, it's slightly easier to do it in maybe the higher species. You can do more multiple administrations in a hollow fiber infection model. There are limits to how much volume you can give a mouse and how many times you can give needle sticks. And certainly it gives you certain advantages when you're trying to study combinations. So let's have a start with how we can use these models to study PK/PD and why there might be an advantage to using an in vitro model to do this. So this example here, it was developed during the development of the combination of avibactam, which was being developed with aztreonam. So avibactam is a non-beta-lactam beta-lactamase inhibitor, and it is we wanted to understand the PK/PD of the avibactam in combination with the aztreonam. So avibactam itself has no activity, and it's actually preventing enzymatic reduction of the activity of aztreonam. But using the hollow fiber system, it allowed us to really maintain the exposure of the aztreonam while varying the exposure of the avibactam to really understand that PK/PD relationship. Another example I'm giving here on the right was where during the development of ceftaroline, and really the take-home message from this is, you can see on the right here, there was a number of different isolates, so MRSA isolates that were investigated in the in vitro model. And if you can imagine that all of this comes from dose ranging for each of these isolates to actually determine this relationship and try and give us an understanding of actually what exposure measure relates best to logarithmic killing. Trying to do this, all of this within animal models would have used a lot of animals and certainly given us a lot more variability. So let's move on to studying resistance. So it's not only sufficient just to look at that impact on bacterial burden. We also want to know what's going on with the resistant population. So we need to understand this profile as well. And so just as a reminder, we believe, the PK/PD relationship with resistance takes the form of this inverted U. So you have some population of resistant bacteria in your initial population. As you apply drug pressure, you'll likely get an increase in this population where your drug concentration isn't necessarily sufficient to overcome the resistant bacteria, but is sufficient to overcome the susceptible bacteria. But if you move forward with this relationship, you'll get to a point where you can optimize concentrations to stop the real growth of that resistant bacteria in the first place. So we need to understand this as well. So how we do this in the hollow fiber infection model is typically what we do, both in the animals and in the hollow fiber is we generally, our main outcome is looking at total bacterial counts. So we'll take our, bacterial sample from the hollow fiber or tissue from our animal models, and then we'll measure bacterial counts normally on drug-free agar plates. When we're looking at resistance, what we're doing is looking at those bacterial counts on drug-containing agar plates. So that gives us an assessment of the resistant population that's growing. So we'll tend to do that on different multiples of MIC with those drug-containing plates. And other measures that we also have a look at is, has the overall MIC shifted at the end of the study? And sometimes we'll also take some of those colonies and retain them and do some whole genome sequencing and look for whether there's some molecular changes. So an example of this in use is here with this example of tebipenem, where we looked at the relationship between tebipenem exposure and both bacterial killing and resistant suppression. So in the red here, you have the total bacterial population, and in the blue here, you have that resistant subpopulation. So it's the same data, but here this is grown on drug-containing plates, and the hashed lines show the pharmacokinetics. So really the take-home message here is you might have drawn one conclusion by just looking at total bacterial population. So you can see Q12 dosing and Q8 dosing is quite good at killing bacteria, but actually you're wanting to go to the much more frequent dosing to really suppress that growth of that emergent resistant population. So moving on now to combinations. The hollow fiber gives you a bit more flexibility in being able to study combinations. So this example was taken from looking at dosage regimens of Astraeanam now given in combination with ceftazidime avibactam. So this was done because for a long time Astraeanam avibactam was in development, and actually people were needing to use something for patients who were infected with pathogens that expressed an NDM, which were, where Astraeanam was no longer efficacious. So at that time, ceftazidime avibactam was already available. So work here was done in the hollow fiber. So remember, at this stage, we understood the kinetics of both Astraeanam and ceftazidime avibactam, but it was important to know, how do you give this in combination? So a number of experiments were done by these investigators to look at that combination, the doses, and also the infusion types. And what's shown here is that if you look at the growth control, Astraeanam monotherapy, ceftazidime avibactam monotherapy, you're seeing no effect. You're still seeing growth of bacteria. But actually, if you give the combination all as a 30-minute infusion, you're still seeing no effect. But here you can see with a 2-hour infusion, you can see an effect, and also with a continuous infusion. So this allowed the investigators to propose how you might want to dose these two drugs in, or I guess it's 3 drugs, but 2, I guess, products in combination. Some other work was also done to look at whether you stagger the dosing of the Astraeanam and the Cazabi, or do you give it all together, and that data is not shown here. Another example here is from the development of flumoxef and amikacin for the treatment of neonatal sepsis. And this is a very detailed graph, and absolutely the take-home message isn't to look at the detail here, but to just show the complexity of experiment that you could do in a hollow fiber, which would have been very, very difficult to do in an animal model system. So this actually was all started off completely in vitro, looking at, a number of different drugs were looked at in checkerboard systems and time kill systems to actually trying to hone down and refine what 2 drugs could be given in combination for the treatment of neonatal sepsis. And what this kind of design allowed the team to do was have a look at a 4x4 matrix, fully understanding that combination. So it tells you a lot more than you can achieve in a checkerboard system, because now you're looking at, a time changing, and also you're able to look at emergence of resistance. So what was actually concluded from this was that the combination of flumoxef and amikacin achieved greater bacterial killing without that emergence of resistance to either compound. So this would have been very difficult to understand just from a checkerboard study or just from animal studies. So the other thing that hollow fiber can give you is much more flexibility around that PK. And so using that last example that I gave you, here humanized PK was used, but it was a humanized PK specifically from neonates was used. So a population model based on neonatal PK taken from clinical studies was taken to recapitulate that data in the in vitro setting. And I guess what I'm showing you here with these PK profiles is you can see neatly how this is the model data from the hollow fiber measured PK following what it should have looked like as a simulation, is you can get quite accurate detail of what that PK looks like. There are humanized animal models, but actually to try and recapitulate what humanized PK looks like, you have to alter the dosing quite significantly. Sometimes you have to alter the renal output in the animal, and it's never quite as accurate as you can see here. And it's also, again, a lot more difficult to achieve. Another example of other, we can look at plasma, but you can also look at, modeling the PK at other sites. So this example here, PK was actually modeled based on human cerebrospinal fluid concentrations, where this was having a look at the impact of meropenem and tobramycin. And again, this was for clinical neonatal meningitis. And actually, one of the conclusions from this set of experiments, this went hand in hand with some rabbit experiments, was that actually, whilst you could model CSF, you needed to use an artificial CSF medium to better correlate to clinical outcome. And that comes on to a later point I make, that understanding what medium you use in these models is also important. So going on to those channels, as well as selection of media, there are problems with the hollow fiber system. Sometimes you have sticky compounds that bind quite well to plastic that you have to overcome. There's a real risk of contamination in these models, especially with the long duration of the models. Once you get small contamination, your whole experimental system can go down and you have to start again. And that's, can happen more easily than in an animal model. They're not cheap. Actually, sometimes it would be cheaper to do these things in animals because the cost of the hollow fiber cartridges themselves is quite significant. And then on top of that, you use very large volumes of media. And then you have some issues with the strains you select. So it isn't, completely that any strain can be taken into the hollow fiber. Some strains can produce quite a lot of gas, and that can be problematic in your system. So to conclude, the dynamic models such as the one compartment and the hollow fiber, they're key tools for translation of PK/PD of antimicrobials. My own personal opinion is that still at the moment, we should consider them in use along with animal models. I think animal models still give you that further information, what's going on, what happens at the site of action, and some impact of the immune system. But certainly there are a lot of advantages of using the hollow fiber for some of the reasons I've explained throughout the talk. So thank you.
Thank you so much, Shampa. So we will move straight ahead with the next speaker of today, Santiago. Just as a reminder, well, I think you probably all know, but within the AMR Accelerator in general, we do a lot of focus on development of tuberculosis regimes and novel therapeutics. Santiago will talk about his own experience in setting up a hollow fiber model, but then specifically for TB, basically from scratch. Of course, there will be some challenges shared, so we're really looking forward to your experience in Santiago. Hello.
So, yes, thank you for inviting us to present the work that we have done under the ERA4TB consortium on the implementation of the hollow fiber system for tuberculosis. And actually, I would like to explain our challenges from scratch on how we were doing this. But before, I would like to set the scene of the ERA4TB context. What we are doing is drug development, and we know that drug development is a very long and costly process with high attrition rate, right? We have a lot of molecules in the very beginning, in preclinical phases. And when we are moving into the development pipeline, we're getting into clinical phases, phase 1, phase 2, the number of molecules are dropping off. and the cost increase and also the timing. So it is actually very important to understand as soon as possible, early in the development process, which molecule is going to become the next drug. And this is actually quite relevant for tuberculosis, that now we have a rich pipeline of new TB drugs at different stages of development, from preclinical to clinical. And in ERA4TB, we are actually changing this paradigm. There was a traditional approach of developing one drug at the time and then combining them. This was taking very, very long, but as you know, TB therapy is based on combination of drugs. So the new paradigm, the new switch is to actually test these combinations from the scratch. But in this scenario, the question that ERA4TB was doing is how can we understand which novel drug combinations to move forward and how can we inform those future clinical trials. And our approach is doing this, generating data from different preclinical, clinical spaces, and integrating this with modeling and simulation. Just to put you a little bit in context, where ERA4TB lays is in the preclinical space, from candidate selection to the clinical space, phase 1 safety. But we work in collaboration with Unite4TB, another consortium under the IHI accelerator program. And actually, we are very well interconnected. So we actually inform how to optimize therapies in the clinic, and the data generated in Unite4TB feeds back to ERA4TB to improve our translational tools. And this translational approach is focused on two aspects. One is capacity creation, in which we are developing novel assays and tools to understand this correlation from the preclinical to the clinic. And also we are progressing individual molecules provided by the pharmaceutical companies and associated partners, and trying to identify how to best combine them in novel TB regimes. But the question again is, how are we selecting these regimes to progress? So one of the many goals of ERA4TB is actually to define this rational progression cascade in which we can inform drug combinations from in vitro to clinic, and to understand this translation. And ERA4TB is focused on, broadly speaking, on in vitro, in vivo, and clinical trials, and we are dealing with in vitro part. We have been developing different tools at adding complexity for MICs, time-kill assays, and especially the hollow fiber system. And to put a little bit the hollow fiber system in context, I'm gonna briefly speak about some things from the MIC and time-kill assays. So MICs is what is typically used in screening campaigns, right? It has a high throughput capacity, and we typically run single shot screenings, but then you need to define for those hits selected their MICs. We do this in 96 well plate here with increasing concentrations, and there is a clear cutoff between grow or no growth. And we can add different regions to actually make easier to identify this cutoff. In this case is the REMA assay, blue inhibition, and pink is growth. Then when we move into combination studies, we use the checkerboard assay. Typically is a 2-dimension with a concentration gradient of drug A in this direction and B in this direction. And depending on the profile, we can assign synergism, no interaction, or antagonism profiles. This is actually quite work consuming and can be optimized using a diamond methodology that basically from the checkerboard plate, we are selecting only the dose response of drug A, the dose response of the other drug, and the equipotent drug concentration of both of them. And just by testing this, we can similarly estimate synergy, antagonism, and even test it a high order. It's not just pairwise, but more drugs in the combination. With MIC determinations, we are getting as an output drug potency in micromolar or micrograms per milliliter, and we have a high throughput. But we have an important limitation, that is single time point and based on growth inhibition. And standardization can be done. We have the UCAS, but this is a very cumbersome methodology and it's difficult to implement across different institutions, although in ERA4TB we are actually working on implementing this capacity. Moving into time kill assays, this is giving us a bit more information. So basically we have a flask in which we can take at different time points samples, and by doing dilutions and plating 100 microliters in a plate, we can estimate the CFUs. The problem with this is that for tuberculosis, the readout is long and we have a low capacity. I mean, you can sample maybe 30 samples each operator. It's very time consuming. In ERA4TB, we are actually improving this, and we demonstrated that just instead of plating 100 microliters, you can plate 2.5 microliters, which you increase by a lot the throughput of the assay, and you can get equally valid results. And actually, we standardized this across several institutions, and we demonstrated that with protocol standardization, we can get consistent and robust data. And additionally, we are coupling these assays not only to CFUs, but to other different biomarkers, phenotypic biomarkers like the CFUs spoken, but the most probably number. This is a liquid outgrowth phase that can detect a subpopulation of bacteria that is not detected by CFUs, which is giving us further information on the potency of the drugs or the combinations. And also we can add molecular biomarkers like the MBLA or the RS synthesis ratio that is giving us information on the physiological states of the bacteria. So as I said, time kill assays, they are giving us longitudinal information. We get information on killing rates, and also we can get information on regrowth on environment or resistance. But there is a big problem, as Professor Shampa has mentioned, is that we cannot modulate the drug concentrations. We add the drug at day 0 and that's it. And this is a problem because we can identify synergistic partners that they have similar PK profiles. So the synergy we identified by checkerboards or time kill assays in theory could be maintained. But if we identify synergistic partners with discordant PK profiles, this is not going to be translated into the clinic. So the question is, how can we experimentally handle this modulation? And here is where the hollow fiber system and the dynamic PK/PD models actually enter into place. As in the previous talk mentioned, the hollow fiber, what it allows is to actually mimic a human PK profile. We can take a PK profile at the site of infection, being blood, lung, wherever it is, and mimic in vitro. And we can play with doses that are safe and to understand what is the efficacy of these doses against different bacterial pathogens. And this can be linked to different biomarkers, as I mentioned before, for time kill assays, or whole genome sequencing, etc. And with this, we can actually play with the PK profiles, right? We can simulate oral administration once a day, twice a day, 3 times a day, IV, and each one of those is going to have a different efficacy and a different toxicity. Then if we couple the hollow fiber system to mathematical modelings, actually we can predict which posology could be the more optimal one. And the hollow fiber is very nicely fits in this space between the preclinical and the clinical phases. So with PK data from phase 1, we can actually simulate in the hollow fiber different scenarios that can inform actually development of phase 2 trials. But how does it look like? So this is a hollow fiber system. You see here, this is the cartridge, and importantly, in the hollow fiber, we have also 4 different ports. As mentioned before, if you see the cartridge, this is a section, you have many fibers. So there is an intracapillary space, so what is inside the fibers. And then we have an extracapillary space, which is where the rest that is contained in the cartridge. So the bacteria, they are actually trapped in this extracapillary space. And for you to see an idea, this is an imaging of one of these fibers. So the extracapillary space would be here on the outside, and this is the intracapillary space where the nutrients and the antibiotics are actually moving. And these fibers, they actually have a small pore site that allow distribution of antibiotics and nutrients and waste, but not transfer of the bacteria. So we actually can trap the bacteria in this extracapillary space. And how does it work? This is a representation, right? This is the cartridge, and then we have a central reservoir and different other reservoirs for diluent and waste. And we couple this to different peristaltic pumps. Here, as you see, again, this is the extracapillary space where the bacteria are, and the intracapillary space that we measure in these pores, S1, S4, is where the nutrients go. So basically, what we are doing is running media through the system, getting an equilibrium. At some point, we can actually inject the drug, and then by playing with different ratios of the pumps and diluting the central reservoir, we are able to actually mimic these human PK profiles. So based on all this, one strategic decision of the ERA4TB consortium was to implement this methodology for tuberculosis to increase this capacity in Europe. Just an historical background on the hollow fiber. The first description of hollow fiber dates back from 2001, and it was used to evaluate PK/PD parameters for a certain antiviral drug. It was only until 2004, 3 years later, we have the first report for tuberculosis, in this case moxifloxacin. And then in 2015, the hollow fiber was endorsed, system for tuberculosis was endorsed by the European Medicines Agency as a valuable tool in the development of novel TB drugs. This endorsement actually had a huge impact. This is a publications on hollow fiber over the years. And you see that around 2015, there was a strong increase in publications, although this tendency is actually, except for this year, is actually going down. So this is a problem that we are seeing, that in the hollow fiber, in the reporting of the literature, that is somehow being less used. Another thing from the European perspective is that despite almost 25 years of its report, the main capacity is in the USA. So in Europe, we have very, very limited capacity. Here we are a few of the groups that are working, but we don't have enough built-up capacity on the system. So this was strategic for the ERA4TB consortium as well, to create this capacity in increasing in Europe. And actually, the hollow fiber has a very high translational value. It has been used in regulatory approvals and submission for different compounds in different pharmaceutical companies, but somehow this gets stuck into the industry and not really transferred into the academia. And well, when we were trying to implement this at the University of Zaragoza, we actually started from scratch, trying to look into the literature, and we identified that there was a systematic lack of information on essential parameters that were very important to actually reproduce this data. In 2021, there was a seminal publication, a review, in which they were doing an analysis of how these methodologies were reported and identifying a lot of gaps in the reporting of the methodology. And they actually proposed a lot of recommendations to improve this. We actually obviously contacted with them at the time, and we started talking, and finally we did a collaboration, and we have written a practical guide on how to use the hollow fiber technology. It's currently under review, hopefully it will be out soon. And the idea of doing this guide is to actually facilitate the access of the hollow fiber technology, and that new people trying to come and implement this in their laboratories, they don't need to suffer what we actually did at the University of Zaragoza trying to implement this. But how was our approach for this implementation? So the first challenge, as I said, is the lack of standards in the field. So we actually try to go very deeply and in detail with protocols and critical parameters definitions, and actually a rigorous reporting. So our work could be then used for it's a robust data that could be used for replication and to build up to future activities. We also put a strong focus on personnel, qualified personnel. It's a quite complex technology. You need to properly train the people, and this training and this know-how is really, really important for the maintenance of the methodology. And actually, we actually did implement some training exchange with some collaborators in both directions. We need to address the complexity of the hollow fiber, and especially if you want to meet the requirements of the people interested, asset owners, or someone that wants to develop a combination. So continuous communication and experimental design is very important. And also, just to address that this is not a systematical process, because every drug is different. As Professor Shampa said, there are problems with binding to the plastic. Depending on the physicochemical properties of the drugs, it's more easy or more difficult to work with the drug into the system. So every experiment has to be properly customized for this. With all this, we actually published an initial work in which we are defining all these criteria. It was spearheaded by these 3 people from our lab. And one of the important things that we actually identified is drug fiber compatibilities, reporting this. Because what we found is that drugs that are being used, it was not demonstrated actually this proper compatibility. Here we have examples that, for example, linezolid in cellulose, the dotted line is what we are intended to mimic, okay? And we have the concentrations in the intracapillary space, which is the central compartment, and the extracapillary space where the bacteria are. And for linezolid, we can actually properly reproduce the levels. In the case of isoniazid, in the intracapillary space, so what we are programming in the pumps, we are actually able to mimic the profiles. But what we observe is that what the bacteria are actually seeing is lower concentrations of INH in this setting. So you need to understand this and then remodulate your pumps to be able to properly reproduce this, because otherwise you're going to have erroneous information. And then we have challenges, for example, the laminate in polysulfone that actually is binding to everything, and we are not able to reproduce these profiles. So actually, building up on this, we have created a hollow fiber TB assay toolkit in which we have tested many of the common anti-TB drugs and reported where are their availabilities. This paper is under review and hopefully will be out soon. Again, with the idea that this is out for the community and we can speed up the development process. And how do we do the hollow fiber system for tuberculosis in ERA4TB? In 3 phases. The first one is the drug fiber compatibility test. We have 3 main types of fibers, polysulfone, PVDF, and cellulose. Then we do a phase 2 in which we actually do PK/PD experiments against the virulent H37RA. And then we confirm all this data with the virulent RV or whatever clinical isolate of interest. And importantly, at every step, we are actually measuring the concentrations outside and inside the cartridges to make sure that we are reaching the levels and the exposures that we are targeting. And we integrate all this with modeling and simulation, both for the design of the experiments and also to data analysis. So at the University of Zaragoza, we have implemented what I believe is one of the largest capacity in Europe, especially in level 3, where we can handle up to 12 cartridges. One of the problems that it was mentioned in the previous talk is contamination. And with tuberculosis, we are actually handling this because we actually work all the time with the cartridges in a biosafety cabinet. So all the manipulations, and this is a requirement due to the biosafety level of Mycobacterium tuberculosis, are done inside the cabinet. And especially the training of the scientists is very, very important. Following on our quest to try to improve robustness of all this, we are actually going through a process, a certification process, ISO 9001, that we expect to have in February 2026, so we can actually ensure that our data are robust and are meeting regulatory and industry standards, but also with all the scientific rigor of the academia. So just to conclude, some opportunities and risk of the hollow fiber system. The opportunity is, as it was mentioned before, there is a global trend of reducing the use of the animals, and actually in vitro systems like the hollow fiber are very valid and useful to fit in silico models that are more and more being accepted by regulatory agencies. And then some risk that this is specific maybe for us, for ERA4TB, in which we have the risk of losing the capacity that has been created under the ERA4TB consortium. And this is especially due to lack of specific schemes for tuberculosis at the European Union level to sustain these investments. So here we have opportunities in that there should be specific funding schemes by the European Union actually to consolidate this investment and to make Europe a reference on this as well. But events like the one today organized to make awareness and diffuse of these campaigns with hollow fiber are very important, and I hope they can actually help to create a robust hollow fiber system community, not only for tuberculosis but also for PKPD in general, of antimicrobials, because it's really important to actually share this knowledge and to distribute to everyone. To finalize, just to acknowledge, this is my team at the University of Zaragoza, and in red, the people working in the hollow fiber. And we do this in tight collaboration with the Institut Pasteur in Lille and with the CNR in Rome. The work we are doing under ERA4TB for the in vitro, there's a lot of people working in here that would like to acknowledge for all these institutions. And again, in red, the people working on the hollow fiber. And finally, to acknowledge all the ERA4TB consortium that has been critical to implement this technology. Thank you.
Thank you very much, Santiago. Very interesting presentation. I'm sure there will be some questions. Very exciting update from your side. Thank you very much. We'll move now to the third speaker of today, which is Alasdair MacGowan. I'm sure that many people know you. So you will move a little bit further into the same discussion, of course, on PKPD, but more with a focus on prevention of resistance.
So thank you very much, Frederick, for being the slide man or the PowerPoint man. I was having a bit of trouble with the with my IT earlier on, so that's why we're doing it like this. I'm going to really build on what the previous speakers have been talking about, because they've covered a lot of the really important points, both in general in terms of how you would set up and think about these dynamic in vitro systems, and also some of the practicalities. If we just move to the next slide, that really shows my disclosures. And like Shampa, we work with a lot of different people in public-private partnerships, sometimes in entirely publicly funded projects and sometimes commercially. And I suppose the key potential conflict here is that I'm a member of the UCAS steering committee, although I do have some involvement in laboratory testing of antimicrobials in the UK. And the next slide is just a little bit of a confession, because although this is called hollow fibers, in fact, I'm not going to show you any hollow fiber data at all. It's all dilutional models. And as Shampa has very nicely explained those, I won't go into that any further. These dynamic models on the next slide have got a whole range of different functions, which the previous two speakers have touched on. But in terms of resistance, I'm just going to talk about how you might assess the emergence of resistance in terms of what the drivers are and what the exposures might need to be. How you might use known or scaled human kinetics to look at bacterial load in terms of long-term experiments. And I think this is really one of the nice things about dynamic models, is that we can simulate full courses of therapy, particularly with rapidly growing organisms. And also we can study drug interactions. And although with monotherapy, we've sort of got a really, at least to my mind, a relatively clear pathway about what we should be doing with preclinical and clinical pharmacokinetics and dynamics. If we've got two active agents, then I think it becomes a lot more complicated. But certainly these models have got significant contributions to make, whether that is an active agent plus a mechanism blocker, which Shampa talked about, Astraeon and Avibactam. The couple of references on these slides are to also to inhibitors, beta-lactamase inhibitors or efflux pump inhibitors, if you want to dive into that. There are other literature out there as well, but also two active agents. And I'll come back to two active agents and resistance. And also there is a hope, particularly with the use of whole genome sequencing in terms of the resistant organisms that come out of these systems, that we might be able to make some predictions about what resistances are likely to arise in clinical practice. Although I would absolutely accept that that is rather speculative at the moment. And if we go on to the next slide, then it really shows some of the factors that are important, and the previous speakers have touched on this already, but you know, picking your strains is important. You know, how many do they have? Are there relevant resistances to the drugs or molecules that you're looking at? And I think what is also really important is to have some strains that you can have some cross-validation with other models, whether these are animal models or other dynamic models, so that if you see different signals, you at least have got common strains. You need to make decisions about inoculum size and growth phase. Are you going to use strains that you know have got high mutational frequencies or not? You know, are you going to do 24-hour simulations, or are you going to simulate 10 days of treatment? What is the drug stability in your system? The classic example of this are beta-lactamase producers, which will digest the beta-lactam when the inoculum is very high, and therefore you don't get the drug concentrations you think, but you might also have binding issues. You need to think about the concentration profile. Is that blood? Is it urine? Is it CSF, as Shampa said? Or is it ELF concentrations? All these things are possible. And how are you going to measure resistant populations? Population analysis profiles is the common way, or are you just going to sequence random 10 strains out of your model? I think it's also useful to think about comparators that are used in clinical practice, if you can, in the situations that you're looking at, because that gives you some grounding to compare your new molecule with, if it is a new molecule, or the question that you're asking about an older molecule. And if we go on to the next one, I just want in the next series of slides show you some data from some EU-funded programs, seeing this is an EU-funded session. And this is some work that we're doing at the moment in GNA now on enteric pathogens with gepatitacin. And this ATC strain, there's published data with gepatitacin both in animals and in vitro systems. So we elected to start here to see if we could show in our system what the driver was, AUC-MIC, that's what the published data says, and what the size of those are for static and kill effects, which are broadly similar to that data published by Brian Van Scoy. So that's nice. We're sort of rooting our findings in something. And if you go to the next slide, it also shows you the emergence of resistance. And as Shampa showed you, we've got this inverse U shape, which was described about 25 years ago, or a bell shape, if you like. And if you just remember that the static effect is around about 45, if you look at the AUC-MIC ratio and look at 45, you'll see that most of the emergence of resistance in terms of population profile changes is happening below that, for the reasons that Shampa explained. This is true on the next slide as well, which is another molecule that we've looked at in GNA now, NOSO502, which is an antimicrobial peptide. And I'm not showing you the response curve here. I've just put it in as a dotted line, what the static effect is. But again, you can see it's the same phenomena where the maximum emergence of resistance is occurring around those static effects or just under. And this is almost a universal truism. I know that if I say it's a universal truism, the next molecule we look at will be entirely different, but we could go back and look at tens of molecules that we've looked at in our system, and it would probably show this pattern. The next slide just illustrates the fact that you can try and work out what the driver is for emergence of resistance. So here we took AUC-MIC ratios with NOSO502 that we knew were going to maximize emergence of resistance. So we just fractionated that by giving the drug once a day, twice a day, or 4 times a day. And as you can see, the emergence of resistance is pretty much the same in these situations, implying that AUC is going to be the driver here for us. If we move on, I'll show you the final example from this EU-funded data, which is a monobactam, similar to Astrionam, AIC499, and you've got the exposure response rate curve, on one side, comparing the time over MIC to log drop. About 50% is the time over MIC for static effect. And then we've got the population analysis profiles on the right-hand side. The solid line is 24 hours. You can see that's lying under 50%. And then as we go on to 48 hours and 72-hour exposures, the longer we expose it, the more likely we are to see changes in the population profile. And again, this is pretty consistent across a whole range of drugs and a whole range of bacteria. And the next slide shows this graphically with polymyxin, which we've been looking at against carbapenem-resistant Acinetobacter baumannii. And some of you might realize that maybe polymyxin is not the best choice on its own for this particular infection. And maybe this type of data helps us here. You can see that you get a rapid kill at humanized dosing, which is just given on the top there, and then regrowth. But also after about 72 hours with both of these strains, you start to see changes in the population profile. So the organism is able to grow on MIC times 4 and MIC times 8 plates. And that is then continues really up to the 7-day profile. And we've also had these strains genome sequenced, which is shown on the next slide. And I'm not going to go through this in any detail, but suffice it to say that in this particular circumstance, this is really quite complicated. Many of the strains do not have obvious mutations related to their outer cell membranes, and that is, say, different to what you would see or what we've seen with aminoglycosides. Where the process seems to be a lot more simple. And then there's a whole range of different mutations which might be related to wall synthesis, some of which have been described in Acinetobacter, and some of which have been described in other species such as Brucella. So I think this really shows us that our previous thoughts about what, if you like, the underlying mechanisms of emergence of resistance in these models, which we used to say, oh, it's up regulation of efflux pumps, and then we get some target site mutations, which is based on the work of George Drusano, which is now about 15 years old, is probably not correct. And what I haven't shown you with the polymyxin data is that there are differences across the 7 days of exposure. So certain mechanisms are more common at certain times than others. So I think for some agents anyway, this might be really quite complicated, and we're not, I would be first to say we're not really quite sure what the translational value of some of this is. If we go on to two active combinations, which is shown in the next slide. This is work with Klebsiella and fosfomycin. And some of you may know that fosfomycin is not particularly active against Klebsiella because of the presence of FOS-A gene in most strains, which is upregulated and the strains become resistant. So if you look at fosfomycin AUC MICs without a second agent, you'll see that there is quite a significant growth on MIC4 and MIC8 plates. Only the strain in the bottom panel do you see the inverse U appearance, because with strains A and B and the top panels, you actually can't give enough fosfomycin to inhibit the strains. So you sort of get half of the U, if you like. However, if you add meropenem, the combination, you get much less emergence of resistance to fosfomycin. In fact, you can more or less shut it off. And what is of potential interest here, at least therapeutically, is that that is not dependent particularly on the meropenem MIC. There may be a threshold with the meropenem MIC, which is very, very high, but we seem to see an effect whether the isolate is sensitive or resistant to meropenem by a monotherapy meropenem breakpoint. And if we can go to the next one, it just shows some similar interaction studies, except these are 7-day experiments. And I'm sorry that this is all a little bit hectic on this slide. These are all strains of Pseudomonas, which are ceftolozone tazobactam, or at least their MICs are above the breakpoint. And if you just look at the top line of each of these triplicates, that's ceftolozone alone over 7 days, and you'll see that there's numbers and counts in those lines for almost all the strains apart from one in the middle, and that really just implies change in population profile. But when we add fosfomycin, which has got pretty modest anti-pseudomonal activity, or tobramycin, which is much more active, then you can switch off those population changes. And that this kind of data is where the thoughts that combination therapy can reduce emergence of resistance. And then on the next slide, just really finish this section on combinations by showing you polymyxin plus a combination, and this is BV100, which is a bioversus molecule. It's basically an IV formulation of rifabutin, and you can see the polymyxin on its own on the far left, where you get kill, regrowth, and changes in the population profile. Well, when you add the rifabutin, you can suppress the emergence of resistance. You can also actually reduce the total bacterial count, but for the purposes of this, it's probably more relevant in terms of suppressing the emergence of resistance. And then the next slide just shows two phenomena, really. This is with strep pneumoniae and fluoroquinolones. It's quite old data where you look at where we looked at different inocula, 10 to the 6 and 10 to the 8. And unsurprisingly, there's more emergence of resistance with a higher inocula, but also with these strains, and they've all basically got roughly the same MICs. The mechanism of resistance is important. So the efflux strain, you are likely to get less further resistance, if you like, than the PAR-C mutation, and this has been shown by other workers with quinolones and strep pneumoniae, and may be true in other situations as well. And so almost finally, if we go to the next slide, I've just sort of tried to summarize some of this for you. That the risk of emergence of resistance in these models is related to what you're exposing, and say Pseudomonas and Acinetobacter baumannii, you're likely to see quite a lot of emergence of resistance, while with some other gram-positive species that's less. The longer you expose, the more likely you're to see resistance. You've got this inverse U-shape relationship. The pattern of drug exposure might be important depending on the driver. Pre-existing resistance mechanisms are important. High inoculum might be important, particularly say if you're thinking about pneumonia as an indication. And the combinations, of course, can be seen to suppress resistance, but what we don't have in the preclinical models is an idea of what the likely toxicological impact might be in a patient population. There are some clinical correlations to all this, which are shown on the next slide. And I'm not really going to go into this in any particular detail, but they're there for you to have a look at. And none of these data are particularly robust, I would say, clinically, but they do support some of the preclinical conclusions in terms of length of therapy. If you like suboptimal drug exposures, species and inoculum being important. However, Ziddentop at the bottom have done a meta-analysis of combinations and emergence of resistance. Unfortunately, this paper is only out on a pre-publication website at the moment, but they do conclude that there is conflicting and only low-grade evidence that combinations will suppress emergence of resistance. So, at least for my money anyway, there's a real urgent need for some further studies in this particular area in terms of combinations. And then finally, there are some limitations in this whole thing, of course. If we go to the last slide, please, that would be great. And the first is that some and possibly most resistance that one sees clinically is actually by horizontal transfer of bacteria or mechanisms, not by the production or not the production of resistance in target pathogens in infected tissue. We generally look at serum concentrations, but of course there are going to be lower concentrations elsewhere in the body, such as on the mucosa, and that might be important, or actually outside the body. You know, what about low concentrations of antibiotic in sewage or down plug holes or wherever they may be in the hospital environment or even the general environment? As I mentioned in relation to the work on whole genome sequencing, it's not always clear that these changes are going to be clinically relevant. And then finally, I think one of the criticisms of this data is that we don't necessarily have white cells in our in vitro kinetic models, although with the advent of more malignant white cell lines, this may well change over time, and it'd be interesting to see if we're successful in introducing, at least in dilutional systems, white cells. That might be more difficult in hollow fiber. And so thank you for your attention, and I will stop there, and thank you, Frederick, for showing the slides for me.
All right, thanks, Alasdair. I'm glad that it worked out fine with the slides. So what we will do now is maybe if Shampa can also turn on her camera. Okay, we have all the speakers. So first of all, I would like to thank all the speakers for bringing it on time, very sharp, very excellent presentations.